Welcome to the deep dive. Today we're digging into a really fascinating ecological study, one that tries to get a handle on a huge question in conservation. What actually makes an ecosystem vulnerable to non-native species?
Yeah, it's a classic problem.
Mm,
Is it that the welcome mat is out because the environment's already stressed? Or is it just that there's a constant scream of potential invaders showing up?
exactly. And the paper we're focusing on today really tackled this head-on. It's titled Aquatic Pollution Increases the Relative Success of Invasive Species, published back in 2011. The lead author is Jeffrey A. Crooks, working with Andrew L. Chang and Gregory M. Ruiz.
It's a really influential piece of work in invasion biology.
Right. So our mission here is to unpack this study. We want to understand what they were investigating, how they designed this really quite clever experiment to isolate the key factors,
hmm
and crucially, what their findings mean for how we might manage these issues.
Okay, so maybe let's set the stage a bit. The big challenge for, decades really, has been figuring out why some places get invaded more than others.
Yeah.
Is it about the local residents, the native species, diversity, what we call biotic factors?
Or is it something about the physical environment?
Precisely. The abiotic factors, things like pollution or physical disturbance, maybe changes in salinity or temperature.
And we often see, just anecdotally, right, that places like busy polluted harbors or maybe disturbed roadsides seem to have way more non-native species than pristine areas.
Absolutely. Think urbanized bays versus a remote coastline. The difference is often stark. But the question has always been why? Is it just because the ships are bringing more potential invaders to the harbors?
That's the propagule pressure idea.
Exactly. Or is it because the pollution or degradation in those harbors actually makes it easier for the invaders to establish once they arrive? The classic confounding factor problem.
Okay, let's dive into that central problem then. The authors wanted to untangle those two variables. Number one, propagule pressure, basically. The sheer number of invader larvae or seeds arriving.
Yep, the supply, like ballast water discharges in a port.
And number two, the inherent susceptibility of the ecosystem itself, its invasibility, which could be affected by, say, pollution weakening the native species.
Right. In most real-world scenarios, like that busy harbor, those two things go hand in hand. High traffic means high propagule pressure, and usually means higher pollution or disturbance, potentially increasing invasibility. It's really hard to tease them apart just by observation.
So the paper's goal, the objective, was to design an experiment out in the field, specifically using these marine fouling communities in San Francisco Bay.
Ah, yes. Stuff that grows on docks and piers,
Right.
barnacles, tunicates, that sort of thing.
Exactly. They wanted to see if they could manipulate the environmental conditions, the pollution level, while keeping the supply of potential invaders, the propagule pressure, essentially constant for everyone.
And San Francisco Bay is a perfect place for this, heavily invaded already, lots of boat traffic, but also areas with varying water quality.
And the pollutant they chose to focus on was copper. Why copper?
Well, copper is a very common metal pollutant, especially in estuaries and coastal areas. A big source is anti-fouling paint used on boat hulls to stop things from growing on them.
Ah, right?
But it also comes from industrial waste, sewage discharge, urban runoff.
Mm.
It's pretty ubiquitous in developed coastal zones, and it's known to be toxic to many marine organisms.
Okay, so this is where the experimental design gets really neat. How did they actually manage to expose organisms to different copper levels without accidentally changing which larvae could reach them? That sounds tricky in open water.
It was very cleverly done.
Uh-huh.
They used standard PVC panels, just simple
Yeah.
plastic squares hung off a seawall. This gives a uniform surface for organisms to settle on. They set it up using what's called a randomized complete block design. Basically, they randomized where the panels were placed along the seawall to account for any small-scale natural variations in water flow or larval supply. And the whole thing ran for 24 weeks.
But how did they apply the copper differently? Did they, like, paint the panels?
No, and this is the crucial part, they used pulsed exposure. Periodically, about every 4 to 6 weeks, they'd retrieve all the panels.
Okay.
Then, for just 72 hours, so only a very short period, less than 10% of the total experiment time, they placed the panels into separate buckets containing different concentrations of copper sulfate dissolved in seawater.
Ah, I see. So most of the time, the panels were just hanging in the bay, exposed to whatever larvae were drifting by naturally.
Exactly. After the 72-hour copper pulse in the buckets, the panels went right back to their original spots in the bay. This meant that the entire natural community of larvae floating in the water column had equal access to all the panels, regardless of which copper treatment they received during those brief pulses.
That's clever. So the supply of invaders, the propagule pressure, was statistically independent of the stress they were applying. They successfully separated the two variables.
Precisely. That's the key innovation here.
And what were the copper levels they used?
They had 5 levels. A zero copper control, which was just seawater in the bucket.
Right.
Then 15, 50, 100, and 200 micrograms per liter of copper. Now, it's important context that levels up to around 50 micrograms per liter are actually found in some polluted marine environments.
Okay, so realistic levels were included.
Yes, and the 100 and 200 levels were sort of pushing it, representing more extreme pollution scenarios, perhaps near an industrial outfall or something.
And after 24 weeks of this cycle in the bay, short pulse in a bucket, back in the bay, what did they find living on these panels?
Well, they identified a total of 44 species or higher taxonomic groups, which they broke down into 16 natives, 14 confirmed exotics, and another 14 cryptogenic species.
Cryptogenic meaning they couldn't be sure if they were native or exotic.
Exactly. Their origins are unknown or uncertain. It's common in these well-mixed port environments. Bryozoans, those little colonial filter feeders, were apparently the most common group overall.
Does having that many unknowns, the cryptogenic ones, muddy the waters for comparing natives and exotics?
It's a fair question. It's a reality of working in these systems. But the authors were careful. Their main analyses focused specifically on comparing the responses of the species they could confidently classify as either native or exotic.
Yeah.
But they still had a solid data set of 30 species with known origins to work with.
Okay, that makes sense. So the big reveal. After 24 weeks, what was the difference between the panels that got the copper pulses and those that didn't? Did the stress matter?
Oh, it mattered hugely. The difference was really striking. Let's look at the native species first.
Okay.
The total richness, the number of different native species found across all panels in a treatment group, declined by more than 40% as copper levels increased.
Wow, 40% just from the short intermittent exposures.
Yep. It was a really strong, statistically significant negative relationship. Basically, the more copper they pulsed, the fewer native species could successfully establish and survive on those panels. The R-squared value was over 0.9, meaning copper concentration explained almost all the variation in native species loss.
That's staggering. A massive hit to local biodiversity.
It really is. Now contrast that with the exotic species.
Well, I guess they didn't care as much.
Pretty much. The total exotic species richness did not change significantly with increasing copper. The trend was almost flat. Copper concentration explained very little of the pattern for exotics. The R-squared was only around 0.35.
So the statistical relationship just wasn't there for the invaders.
Correct. And crucially, the slopes of those two response lines, the sharp decline for natives versus the flat line for exotics, were significantly different from each other. It's clear evidence of a differential response.
So for natives, copper was like a major barrier, but for exotics, it was, what, barely noticeable?
Essentially, yes. In fact, they found something even more telling. 6 of the 14 exotic species were actually found more often on the plates with the highest copper levels compared to the zero copper controls.
Wait, they did better with more pollution.
It appears so, or at least they were relatively more successful in those high-stress conditions, possibly because their native competitors were knocked out. They mentioned specific examples, some invasive bryozoans like Cryptosola and Electra and Atunicate Siona. These guys seem quite tolerant.
And did this change the overall look of the communities?
Absolutely. Copper caused significant shifts in the whole community structure. For instance, native barnacles, which normally dominate space on these panels early on, decreased significantly with higher copper. That opened up bare space.
Which the copper-tolerant exotics could then colonize.
Exactly. The pollution actively restructured the community in favor of the invaders.
Okay, this feels like the core finding. It really seems to confirm that pollution can act like a secret weapon for invasive species, even when you hold the number of arrivals constant.
That's the major takeaway. The results strongly support the idea that human-caused changes to the abiotic environment like pollution directly influence invader success, independently of propagule pressure.
So that anecdotal observation about polluted harbors having more exotics wasn't just about ship traffic.
It seems not. Or at least, it's not only about ship traffic. This study provides strong evidence that the degraded conditions themselves play a critical role in filtering out natives and favoring exotics.
Which brings us to that key theoretical idea they discuss, the selection regime alteration hypothesis. Can you break that down for us? It sounds important.
It really is. It's about thinking what environmental pressures species are adapted to. Native species in, say, San Francisco Bay evolved over long periods to thrive in the historical conditions of that bay, relatively low pollution, specific temperature ranges, existing predators and competitors.
Right, they're adapted to the normal local environment.
Exactly. But when we introduce pollution, like copper, we fundamentally alter that selection regime. We change the rules of the game. The conditions are no longer what the natives are best adapted for.
Okay.
Now think about the exotic species. Where did they come from? Often, they've survived a really tough journey, maybe weeks in a dark, potentially metal-contaminated ballast tank, or clinging to a ship's hull, experiencing huge variations in temperature and salinity.
So the journey itself is a harsh filter.
Precisely. The ones that survive that journey are often, sort of by definition, tough, stress-tolerant species. They might be pre-adapted to disturbed or polluted conditions simply because they had to endure similar stresses during transport.
So they arrive in a polluted harbor, and while the conditions are tough for the natives, for the invaders, it might feel kind of familiar, or at least manageable compared to the journey.
That's the essence of their hypothesis. The pollution stress that harms the locally adapted natives might not be as detrimental to the exotics that have already been selected for tolerance to harsh artificial conditions. The pollution inadvertently selects for the invaders by removing their competitors.
That really reframes how we think about invasions in degraded areas. It's not just that the natives are weakened, it's that the environment is actively favoring species with a different kind of life history, the hardy global traveler.
Yeah, and remember, this experiment started on clean, bare panels. That mimics situations like after a disturbance, maybe dredging, or a pollution event kills off existing life, or even just newly installed docks or seawalls.
Right, early stages of colonization.
And the study showed that in those early stages, the degree of invader success was directly linked to the level of copper stress. More stress, relatively better performance by the exotics compared to natives.
Now, you mentioned this was a field experiment, and those are always challenging. Did the authors note any limitations or complications in their results?
They did, and it's good practice to acknowledge them. They pointed out two main things. First, there were significant block effects, meaning that even with randomization, the specific location of a panel along the seawall still mattered. Panels in slightly different spots ended up with somewhat different communities, likely due to small natural variations in water currents carrying larvae or maybe light levels, things like that.
Yeah.
Just reminds us that even over short distances, nature isn't perfectly uniform.
Okay, that makes sense. What was the second limitation?
This one is perhaps more intriguing. They found an issue with their control group specifically, the panels that were handled exactly like the others, taken out, put in a bucket, put back, but without any copper added to the bucket water. The zero copper buckets treatment.
What was the issue?
Well, the communities that developed on those zero copper buckets panels actually looked more similar to the panels that received intermediate levels of copper, like 15 or 50 colmols, than they did to a theoretical true control that was never handled at all.
Wait, just the act of putting them in a clean bucket made them look like they'd been slightly polluted. That's weird.
It is weird, but it suggests something important. It implies that the physical handling itself, taking the panel out of the water, the change in environment even for just 72 hours in a bucket, might have acted as a form of stress or disturbance.
Like a physical stress rather than chemical.
Exactly. And this minor disturbance might have slightly disrupted the native community, perhaps reducing competition just enough to allow some less competitive species, maybe including some exotics, to get a foothold. Similar to what happened under low chemical stress.
Yeah.
Might be an example of the intermediate disturbance hypothesis.
So even trying to create a perfect control introduced a small effect. Field ecology is tough.
It really is. But it's important to stress that despite this nuance with the control, the overall pattern of natives declining sharply with increasing copper while exotics remained tolerant was incredibly strong and statistically robust. The main conclusion holds firm.
Okay, so given that strong conclusion, what's the big management takeaway? If pollution helps invaders independently of how they arrive, what should managers be doing?
The message is pretty clear. You have to tackle both problems together. We spend a lot of effort on vector management, trying to stop invaders from arriving in the first place, like treating ballast water or cleaning ship hulls.
Right, controlling the supply.
But this study really underscores that reducing propagule supply alone might not be enough if the destination environment is degraded. You need to simultaneously work on improving the environmental conditions, reducing pollution, restoring habitats.
Because if you only stop the ships but leave the harbor polluted.
You're essentially creating an environment where any invader that does slip through the net finds conditions that favor it over the weakened native species. It makes the few arrivals more likely to succeed.
And conversely, cleaning up the water without managing the constant influx of new species is also likely insufficient.
Exactly. It's a two-pronged problem needing a two-pronged solution. Vector management and habitat restoration or pollution control need to be tightly coupled. You can't really expect one to work fully without the other.
So, summing up this deep dive, the Crooks et al. Paper provides compelling experimental evidence that environmental degradation, specifically copper pollution in this case, can directly promote the success of invasive species by harming natives more than stress-tolerant exotics, quite apart from how many invaders are arriving.
It really shifted the focus, highlighting how altering the environment itself acts as a powerful selection force.
Okay, so here's a final thought for you, our listener, to consider. This experiment ran for 24 weeks, capturing that crucial initial colonization phase. The authors predicted that ongoing pollution would likely lead to long-term dominance by exotics. But is that initial success guaranteed to last?
That's the question, isn't it? What happens over years, not just months?
Right. What kind of longer-term studies would we need to see if this initial advantage for exotics persists? Or could native species, perhaps the more resilient ones, eventually recover and compete effectively even in moderately polluted conditions once that initial scramble for space is over? Does the long-term community dynamic look different from this early snapshot?
Yeah, understanding that longer trajectory is definitely a key area for future research.
Something to mull over. We hope this deep dive has given you a solid understanding of this important study and its implications for how we view and manage aquatic invasions. Thanks for joining us.
Next time.